Thank you very much, Thibaut and Virginie. I think one of the things I really enjoy about working with Fruition Sciences is very much trying to understand the relationship between how vines are growing and wine composition. I'm very much on the wine side in terms of trying to understand phenolic composition. Most of my research is in the phenolic area, and trying to understand how vines are growing and how that translates into fruit composition and then ultimately to phenolic quality in the bottle. And so much of my research is very much focused on that. So a quick outline of what I'm going to talk about today. I want to quickly introduce phenolic classes, location in the berry, and kind of winemaking sensory considerations. I want to give it to you from my perspective on it in terms of how a winemaker might approach fruit and how that might go back into the vineyard and how you might manage vine growth. Talk a little bit about phenolic development and then ultimately the response to environment. And then managing wine composition in the vineyard is what we're ultimately after. I'd like to be able to look at a vine and see those bottles of wine and really ultimately say, all right, that's a high quality bottle of wine that's growing in the vineyard right now. So quickly, the phenolic classes that we tend to think about are hydroxycinnamic acids, which are important in white wine color, flavonoids, anthocyanins, tannins, catechins, and flavonols. These are all important compounds in wine quality. Anthocyanins, obviously, for red wine color. The tannins, astringency, mouthfeel. Catechins impart bitterness in wine. They're essentially seed-derived. And then the flavonols, not necessarily directly implicated in wine quality, but I think they're great marker compounds for monitoring exposure in the fruit zone. So more fruit exposure, higher flavonol concentrations. And then we have minor contributors like hydroxybenzoic acids, which ultimately are derived from seed tannins and oak cooperage if you do that. So what is the approach that I tend to look for? And when talking to winemakers, you very much get the question, you know, what is this target that we're all after? And I think it depends on who your audience is. Is it the wine review writer? Is it a wine consumer? So on and so forth. And when I was working at the Australian Wine Research Institute, I think this study kind of puts it nicely. This is some work we did with Hardy's Wine Company in Australia. And what Hardy's was trying to do was trying to assess phenolic composition in wine as a function of their sourcing. And so they had a number of vineyards. These are all Shiraz wines. And what the winemakers did was they segregated all these Shirazes according to where they saw the price point of that wine going. So on the far left are the higher price point wines for these Shiraz wines. And then on the far right, the lower price point wines. And the price point variation for these Hardy Shirazes was about from $100 plus down to $5 to $8. So it very much encompassed the Hardy's price point range. And what we did at the Australian Wine Research Institute is we analyzed those wines to try to understand what's going on in terms of phenolic composition. And there was no question we saw some pretty strong relationships. At the ends, the low price point wines, and at the high price point wines, there did not appear to be any kind of phenolic drivers in terms of quality, in terms of what the winemaker was targeting. I think at the high price point, you can almost argue the phenolics are optimized and that the price point had more to do with potential marketing issues and or some other attributes such as flavor and aroma. Lower price point wines, there's all these other issues that we have to deal with. It's not really phenolic related. There's potential flavors again, potentially mouthfeel characteristics as in fullness and through the middle that aren't necessarily phenolic driven. What was interesting was that middle portion of that vineyard, this graph, where tannin concentration, tannin molecular weight, percent of epigallocatechin and pigmented polymer were strongly related to price point that the winemakers perceived, and overall wine color density. And in Australia, they developed this metric for looking at wine color density in red wines as a price indicator, as a price point indicator for wine allocation. But grape growers are smart, so they very quickly optimize color in red wine. Unfortunately, that's not the solution to the puzzle, is it? There's a lot more going on, and that's why the Australian Wine Research Institute was asked, are there other metrics that we can start to look at beyond color to try to optimize price point? The other thing that I'll point out here is that when the vineyard managers were brought into this discussion, they very much saw this relationship where it's actually how those vines were being grown that determined the phenolic concentration. And typically you're dealing with growers who are either quality driven or quantity driven, and that ultimately started to drive phenolic composition in the wine. Just a tabular representation of that information. And what you can see on the right-hand side is correlations. And there's some pretty strong correlations in that linear range of that curve where tannin concentration, degree of polymerization, this percent epigallocatechin, that's a marker for skin tannin. So percent skin tannin is very strongly related to phenolic quality in the overall final wine, okay? And then there's all these color attributes as well. And then polysaccharide concentrations in the wine. Polysaccharides are thought to give fullness to wine, and they're a product of cell wall deconstruction in the grape berry. So as that berry develops, as it matures, as it starts to get softer, you start to release more polysaccharides. And so there's definitely a relationship there. One of the conclusions, if I could go back, you often get this, well, if tannin concentration's driving this, why not just buy a bunch of tannin and dump it into the fermenter, right? That's a reasonable thought. I think the thing to think about is that we're all after balance in our wines, right? So it's not necessarily that these wines have higher phenolic concentrations, is that they're balanced at a higher level of tannin, or they're balanced at a higher color concentration. So yes, these phenolics are higher, but there's something about the matrix of that wine in addition to the phenolics that is allowing that wine to build more structure, okay? So this is a review, I'm sure a lot of you have seen this, published in 1993 by Jackson and Lombard. There's a lot of inputs into wine quality. These are the very much, I think we're all in this room because we all believe that grape composition is everything. And so there's all these inputs, and Thibault and Fruition Sciences spends a lot of time trying to understand how all these environmental factors are ultimately affecting grape composition. And that's what we're trying to do in my research team, is in the vineyard understand what are these environmental inputs and how are they ultimately affecting composition. If we focus in on tannins and color, which I think we're all interested in, it's good to understand when tannins are produced in the grape berry and when color is produced. I think color is pretty obvious, post-veraison. But for the tannins, it's always pre-veraison. So tannins are produced from flowering up to veraison and soon thereafter. C tannins are produced a little bit longer than skin tannins. But then there's this period during fruit ripening where these tannins, quote unquote, mature. And it's really trying to understand what can you do in the vineyard on the pre-veraison side in terms of producing the amount of tannin versus what can you do on the side of post-veraison to affect that maturation profile. Looking at literature, I think this is kind of sums it all up in terms of, if you're looking at phenolics, we tend to think of moderate stress as being a thing that promotes quantity and quality in phenolics. And when I say moderate stress, we're thinking of things as high exposure, sun exposure, lower levels of nitrogen, low soil moisture, moderate canopy size, moderate crop load, low soil fertility, small berry size. I think a lot of the talks today so far have kind of sums it up and kind of are consistent with this in many ways, although we do periodically get thrown curveballs, don't we? It's really about how does that fruit get exposed and how do we manage balance in the vineyard so that we can affect these phenolics positively. So phenolics in the environment, evidence indicates that berry phenolics are dependent upon cluster environment alone. And that's an important point, and I think it's there's some obviously some potential minor components, but for the most part, it's looking at that fruit, understanding the environment that fruit's exposed to, which drives a lot of what the phenolic composition of that fruit's going to be at harvest. Fruit exposure is positively associated with phenolic quality, unless you live in Fresno. That's actually negatively related to phenolic quality. It's because of sunburn and heat. So we'll get into that in a minute. Research on phenolics has definitely focused on phenolics per se. Phenolics reside in the solid parts of the berries, And I think one of the things that historically we've always looked at is how much phenolic material is in the grape berry. But it's important to realize that it's not simply just how much phenolic material is in the grape berry, but as winemakers, you're extracting this material, and so there are diffusional barriers that are in the berry that limit how much phenolic material is going to get out. Simply due to rates of diffusion, there are differences in terms of rates of diffusion, but the material that limits the diffusion of phenolics out of the berry also interacts with the phenolics, and they are great binding sites for phenolics and so on. So there more recent research is showing that we not only need to be thinking about how much phenolic is there, but we also need to be thinking about the diffusional barriers of those phenolics. So this is the exposure conundrum. Full shade, we're taught as viticulturists, full shade is perhaps not a good thing to have in our fruit. Contributes to low sugar, low color, low fruit character and flavor, high green character, higher acids, higher must nitrogen, which is not a bad thing if you're trying to get good yans in your fermenter, increased disease pressure. Full exposure is great if you're looking for higher sugar, higher color, lower levels of methoxypyrazines, lower levels of acidity. One of the potential downsides is lower levels of yan, but you have reduced disease pressure, and there's bottom line increased risk of sun exposure. So leaf pulling is great, but at some point you get whacked, and there's we all have heard of the, we pull a lot of leaves off of our clusters to get the exposure that we want in our fruit zone, and we get sunburn events that happen. I think what we're trying to achieve is a balanced vine, as the talks earlier have shown, but it's also allowing enough shade to protect the fruit, but enough light to produce the fruit character and the phenolic composition that we want. I always like to talk about this project that we did. I used to be a faculty member at Oregon State University, and we did some work with Archery Summit to try to understand some price points on their couple of blocks, $38 bottles versus $75 in terms of what is going on with these phenolically. The winemaker, Anna Matzinger, very much was convinced that the price point was related to phenolics, and she wanted to understand what was going on. So I had a student go out there and measure vine vigor. There were some obvious vigor differences in this vineyard. And on the upper left-hand graph, you'll see this plot of the vigor zones within these two blocks that are being compared. Greener sites were higher vigor, red site, low vigor. And what we ultimately did is we collected fruit samples from these two different blocks, and we looked at the composition of the phenolics in the fruit. The upper right-hand side is a real interesting one in my mind, and that's tannin concentration in the skin tissue. And what this student, Jessica Cortell, was very much able to show was that tannin production in the skin tissue, and this is milligrams per berry, so this is on a unit level, milligrams per berry of tannin was higher in the lower vigor sites. And that translated into pigmented polymer, and then percent epigallocatechin, which is that marker compound we saw in Australian Shiraz as well. So it seems to be something related to vigor. Making wines from these blocks, low vigor to high vigor, you can think of the price point of these wines as increasing as you go from left to right. If you look at the composition of the tannin in the red wine, so we have the ability to analyze the red wine, analyze the tannin in that wine, and determine where that tannin originated from, whether it came from the skin tissue or whether it came from the seed tissue. And what we were able to show in this graph is that as the vigor declined, as the price point of the wine increased, the percent skin tannin increased along with it. What's interesting is if you look at the lower graph, while the percent skin tannin increased, what we found was that the amount of seed tannin contribution to the wines was fairly constant. It didn't change too much. What changed was how much skin tannin was going into that wine system. The winemaker, when tasting these wines, very much looked at the lowest vigor wine and said, you know, that's not exactly where I want it to be. It's got a great phenolic profile, but we're starting to introduce negative flavor characters in that wine, okay? On the far left, the highest vigor sites had the highest proportion of seed tannin, lowest tannin. It's interesting that the winemaker said, while the amount of seed contribution to that wine was the same as all the other wines, I picked up bitterness in that wine. So that astringency was able to mask bitterness in the wine due to that. How am I doing on time? Am I okay? Yeah. So what we did in that, yes?
On that last slide with the high vigor, was the seed very
so the question is that were there differences in seed coat color between the high vigor and the low vigor? No. And in typical Oregon situation is you have green seeds regardless of condition. There are exceptions, but they're dealing with green seed issues typically in Oregon. And they're not significantly so, no. The thing that came out of this is trying to understand how do we control that vigor. Certainly one is what's the cause of the vigor difference, and what we found in this study was that soil water holding capacity was very much related to vigor in the zone, and the prescription for that winery is control your water relations in your vineyard. You can help to restrict the vigor in the vine and get the exposure that you need in that fruit zone. One of the alternative things that you could do is manage the environment around that fruit. And so what we did in year 2 of this experiment is control the environment around the fruit while maintaining a constant vigor. And what we were able to show in this portion of the study was that the phenolics that are driving the apparent quality according to the winemaker are very much exposure related. And so we covered up clusters. And when you cover up clusters, you reduce skin tannin biosynthesis, you reduce color in that fruit as well, even though the size of the fruit is all the same. And so that was kind of interesting. It seemed to clearly indicate an exposure situation. Follow-up experiments in a subsequent years with a different student where we're keeping the light constant but manipulating the temperature. And this is some work working with Julie Terrara at the USDA up in Washington State on Merlot, very much showed that while temperature has a very clear effect on ripening profiles, sugar accumulation, so on and so forth in that fruit, what is not affected so much is tannins. And tannins very much seem to be a light-driven phenomenon. So much like flavonols, okay? So flavonols, very light-sensitive. Tannins appear to be light-sensitive as well, as opposed to temperature-sensitive. So some general take-homes. Phenolics are an important component in wine quality, certainly wine scores, price point. Phenolics change during berry development, including components that influence perception. And phenolics can be managed. I mean, there's a lot of things that we can do to do this. Analytical chemistry has made significant strides in the past decade. We're still on the research side. There are analytical labs in California who do specialize in phenolics. And I think, you know, I'm of the ilk that the more information you get, the better. On the research side, we're definitely working with a lot of wineries and vineyards right now to really understand what is this underlying science of phenolic quality in the vineyard. Skin versus seed tannins are very important. And one of the things that we're looking at increasingly is not just whether it's a seed tannin or it's a skin tannin, but ultimately how active is that tannin. Okay, and so we've developed analytics to measure tannin stickiness or tannin activity. Some people will call it grippiness. And we're understanding and exploring how grippiness is driven by vineyard practices. Finally, some take-homes. Anthocyanins, if you're really thinking about optimizing color in your vineyard, research indicates that temperature is critical. So unlike tannin, which suggests it's a light-related phenomenon, anthocyanins seem to be more temperature-driven, and they tend to peak at about 30 degrees Celsius. And so temperatures in excess of 30 degrees, you tend to start breaking down color in your grapes. So overexposure in warm climates is not such a good thing for red color. And then alternatively, cooler temperatures, you see less biosynthesis of anthocyanins. For the tannins, monomers decline with maturity, and they're less sensitive to skin tannin to change in the environment. I often think of seed as being in a nice little insulated sac, And so they're going to be less sensitive to environmental changes just because there's some insulation around that seed tissue. Obviously, if the seed gets, or the berry gets too small, you're going to ultimately change the development course of that seed material. Question? So, and that's a good question. The question was, the 30 degrees Celsius, is it ambient temperature or is it, what is, you've got a black body there, it's potentially actually warmer than the fruit. And yeah, that's a good point. Actually, Julie Terrara, a lot of this data is based on studies by Julie Terrara, where she sticks thermocouples actually into that skin tissue to try to understand what that temperature of the skin is experiencing. And it's about 30 degrees Celsius at that skin. So not necessarily ambient, it's the thing that the berry is experiencing. With regard to skin tannins, very much sensitive to exposure. Light is important. Quality is an open question. In terms of quality of light, UVB seems to be the portion of the UV spectrum that tannins are sensitive to. And there's evidence for upregulation. There's this thought that if you give a berry a unit of energy, it's going to produce a unit of tannin. And so I think it's important to understand that berries are very plastic, and the biochemical pathways within a berry can be upregulated and downregulated to the berry's whim. And one of the things that we do find in this studies that I've seen and I've conducted is that a berry varietally wants to be at a certain point. And so in this experiment where we manipulated the temperature, cooler cluster temperatures initially reduced the amount of tannin produced, but by second time point, third time point, the berry had the ability to compensate. And so you absolutely saw an upregulation in the biochemical pathways that geared towards tannin production, and that berry ultimately caught up in terms of overall tannin. The other thing that I'm not pointing out here is these diffusional gradients. And we're really starting to understand cell wall much more so than we have. Cell walls are a great binding site of absorption for phenolic material. And so increasingly we're looking at the solvent strength of that wine system. The amount of cell wall material you're throwing in a fermenter can ultimately have an effect on how much phenolic you're able to extract from that system. And that's the environmental effects of cell wall biosynthesis is still in its early stages. And that's all I have for you. Happy to answer any questions. Yes?
In one of your earlier slides, so post-spirating is kind of about immature. Do you know much about that?
We do. I think we know some. So if you look at phenolic maturity in the seed, there's, with regard to tannins, as that grape gets riper, the proportion of astringency relative to bitterness declines, or astringency increases overall with maturity. And so what we see is that the lowest molecular weight tannins tend to disappear more rapidly than the higher molecular weight tannins. And there's evidence that that tannin is getting oxidized. And so that in the seed tissue, overall, the riper that seed is, the more astringent the seed's going to become, as opposed to bitter. So there was a question earlier about green. Potentially, there's some issues with potential bitterness extraction if you have too much green seed. It's an immature seed, more bitterness. On the skin, it's a little bit more complicated. The skin tannins, there's color incorporation into the tannins in the skin. It's still an open question as to the significance of that proportionally to the overall tannin that's extracted. But from a maturity standpoint, no question, more color incorporation in the skin tissue seems to be the driving factor. Yeah, so the question is, with regard to the diffusional barriers, if you have more diffusional barriers, does that potentially limit the extractability of the phenolics out of the grape berry and into the fermenter? And yes, it does. So the diffusional barriers, you have to think about it. If you've done extended maceration before, you've seen that cap and how it degrades over time. You know, by the time you get into very long soak times, that cell mass is breaking apart. And so you can very much think of these diffusional barriers as just opening up and letting go. The flip side of that, however, is that more diffusional barriers, there's more cell wall mass that you put into that fermenter. And so there's more surface available for adsorption. There are more sites of adsorption. So it's not just that there's a screen that's limiting things to getting out of the plant cell, but it's also the surface that is in there is providing potentially more sites for interaction. Does that make sense? Great. Thank you very much.